接下来这个 speaker 是Max Hasmann. Dr. Hasmann, he get his diploma degree in biology in 1982 at Munich. Then he get his PhD degree at the Institution for Microbiology in 1985. And he has been the head of cell culture lab oncology research in Germany from 1987 to 2001. And from 2016, he has worked in the translation medicine scientist in TM appointment, member of EI groups. Today, he is going to give us a very inspiring and interesting talk about the overcoming resistance in HER2-targeted therapy in breast cancer. Let's welcome him.
Thank you very much for the kind introduction. And I would like to thank the organizers for giving me the opportunity to talk in this audience about the progress we have made in treatment of HER2-positive breast cancer and how we try to overcome resistance to HER2-targeted therapies. Now, the story of HER2 targeting actually began back in 1986 when Hasmann et al. Published that patients whose tumors was overexpressing HER2 or had an amplification of the HER2 gene had a worse prognosis than HER2-negative patients. So they had shorter survival times and shorter time to progression times. Now, with the introduction of Herceptin, which was a consequence of this finding, we realized that Herceptin really changed the natural course of HER2-positive breast cancer. And nowadays, HER2-positive breast cancer patients seem to have even a better prognosis than HER2-negative patients. So Herceptin turned this more or less around. In metastatic breast cancer, but also in early breast cancer, there was a big progress. Now, as you can see from these curves, there is still a high medical need because a lot of patients tend to progress after some time of treatment. Of course, the question is, what are the reasons why these tumors after some time progress? And what are the reasons of HER2 targeting or resistance to HER2 targeting? Now, we have identified a number of potential resistance mechanisms in vitro and in preclinical models, and many of them have been tested in clinical trials. This is just a list of the potential resistance mechanisms we have tested. I will not explain them in detail, but I can say that none of these potential resistance markers has really translated into benefit in clinical practice. So in the end, HER2 remains the only marker for patient selection. Now, what can we do in order to improve HER2-targeted treatments? We have basically followed two approaches, and one of the approaches was based on the finding that HER2 overexpression remains beyond progression on Herceptin. Now, this is explained by the fact that the HER2 overexpression is based on the gene amplification, and there is no physiological mechanism to eliminate gene amplification. So HER2 remains overexpressed. And the idea was to create an antibody drug conjugate, so use HER2 as for targeting toxins to the tumor. Now, this looks like an easy concept. However, it is quite complicated because the actual antibody-drug conjugate needs to be tailored to the properties of the target, to HER2. And as you can see here, HER2 is relatively slowly internalized compared to the EGF receptor. And what is also characteristic for HER2 is that when internalized, most of the internalized HER2 is actually recycled to the membrane. So only little HER2 enters the late endosomes and the lysosomes where it would be degraded. And this internalization and recycling mechanism is not changed when trastuzumab or Herceptin is bound to HER2. What does this mean? Only little antibody-drug conjugate would enter the lysosomes and release the toxin. Therefore, we need a very, very potent toxin. And on the other hand, we need an extremely stable linker because most of the antibody-drug conjugate will be on the cell surface where the toxin is not supposed to be released. Now, this is the result of several years of optimization of such an antibody-drug conjugate. It's now known as Kadcyla. The original name was TDM1. And the construct is like it's based on trastuzumab as an antibody. It contains an extremely stable linker, this MCC. And it contains DM1, a very toxic tubulin inhibitor, whose development as a chemotherapy agent was stopped in the 1980s because it was just too toxic when used as a single agent. But for an antibody-drug conjugate based on trastuzumab, it turned out to be really good. And the coupling ratio is about 3.5 to 1. I was told that you are very familiar with the clinical data, so I'm just showing the Emilia results here, which led to the approval, to the final approval of Kadcyla in second-line treatment for metastatic breast cancer. And Kadcyla was first approved in the US in 2013. We followed another approach to improve HER2-targeted treatment, and this was based on more insights into the mechanisms of HER2 activation and also into the mechanisms of Herceptin as an antibody targeting HER2. So one of the mechanisms of HER2 activation is HER2 protein overexpression and the formation of ligand-independent HER2 homodimers. Another mechanism is the proteolytic cleavage of the extracellular domain of HER2. This is also an activation mechanism because the remaining truncated receptor called P95HER2, which contains the transmembrane domain and the intracellular domain, is a very active form of HER2. And it's dysregulated because the regulatory extracellular domain is lacking in this molecule. And finally, it was found out that there is another mechanism of HER2 activation, which is the formation of heterodimers with other HER family members like HER3 or HER1. And those receptors would need to be activated by a ligand, by a growth factor, respectively. Now, when the mechanisms of Herceptin were more intensively investigated, it turned out that Herceptin would inhibit the ligand-independent activation of HER2, and it would inhibit the proteolytic ectodomain cleavage, the shedding of HER2, but it could not prevent the formation of HER2 heterodimers. And therefore, another molecule became interesting again. Now, this slide This slide shows several antibodies that were derived back in 1987 when researchers at Genentech immunized mice with human HER2. And the most active clone, 4D5, was chosen at that time for development of a monoclonal antibody. Now, this antibody, 4D5, is now known as Herceptin. Another antibody called 2C4 was discarded at that time, but it was derived from the very same immunization as 4D5. It was discarded because it was less active on cell lines than 4D5. However, continued research at Genentech in the 1990s found out that 2C4 was different to 4D5. And the difference was as follows. So 4D5, or trastuzumab, Herceptin, binds to subdomain 4 of the extracellular domain of HER2. As you can see, HER2, the extracellular domain, looks a bit like myself seen from the side. So here is a nose. This is actually the domain where the dimerization with other receptors occurs. There's a little beard, and this is the neck. And Herceptin would bind to the neck. And it's important to realize it binds to the side of the neck, not to the throat. And this is the reason why it cannot prevent the dimerization with another receptor. But it can prevent the cleavage of the extracellular domain because it prevents access for the proteases, which would cleave the extracellular domain here. Now, the other antibody, 2C4, or pertuzumab, as it is called now, binds to a different region. It binds directly onto the dimerization nose, as I call it. So pertuzumab prevents the dimerization with other her family members, but it cannot prevent the cleavage of the extracellular domain. So the two different binding epitopes of the antibodies result in different mechanisms of action. Now, when this was realized, people were thinking that the formation of HER2 heterodimers were mainly important in HER2 low-expressing tumors, whereas the shedding and the ligand-independent formation of HER2 homodimers would mainly occur in HER2-positive tumors. This was the thinking at the beginning, and this was the reason why, when pertuzumab was developed initially, we did 5 phase 2 studies in 4 different indications. So you can realize that there was a lot of excitement on pertuzumab at this time because we did 5 phase 2 trials in parallel, and in 4 different indications. They were all in HER2 low-expressing tumors, and the results were quite disappointing. Some activity was seen, but overall there was no reason to go on with a phase 3 study. So at that time, in 2005, Genentech basically terminated the program. We were still different companies, Roche and Genentech, at that time. However, at Penzberg, where I'm working, we had just done the first combination experiments with Herceptin in preclinical models. And we were really intrigued by the fact that the 2 antibodies would have 2 different mechanisms of action which may complement each other. So we did an experiment on a HER2-positive non-small cell lung cancer xenograft model. And as you can see, the single agents antibodies, they could delay the growth of this tumor model quite significantly. However, when we used the combination, we got tumor regressions, and in this experiment, even 3 out of 10 animals were cured, so there was no longer any tumor measurable at the end of the experiment. And we used another xenograft model, a HER2-positive breast cancer model, and here the effect was even more pronounced. So you can see that the single agents could delay the growth of this tumor model for about 2 weeks only, and then the tumors grew just as fast as the control tumors, the untreated control. But when we used the combination of the 2 antibodies, we got tumor regressions. And in this experiment, even 6 out of animals were cured. So this was amazing. We had never seen such a synergy in a preclinical experiment before. And we got really excited. But it was not easy to move forward because there were a lot of questions. First of all, if you have 5 negative phase 2 trials, it's really difficult to persuade anyone to move forward. Then there were some scientific questions like, in HER2-positive tumors, there is a really vast excess of HER2. Why do you think that heterodimers would play a role if HER3 or HER1 is expressed at very, very low levels in these tumors? And then the question, how would you persuade clinicians to use 2 antibodies binding to the very same target? And finally, the business people were thinking, treatment with Herceptin is quite expensive. Who would ever pay for 2 antibodies? And 2 antibodies binding to the same target. So there were a lot of questions, and it was not easy to move forward. But, yeah, and then we also were considering the clinical trial we could do, and this was also not easy because we came to the conclusion we can only do a study in patients whose tumors progressed under Herceptin, because Herceptin was standard of care, and this is a challenging situation. So we did another experiment and treated this breast cancer model, KPL4, with Herceptin until tumors progressed. And then we added pertuzumab, and you still can see the synergy. Although we had no complete remissions here, this was mainly due to the fact that tumors were already relatively big when we started to treat the mice with the combination. Nevertheless, it showed that the synergy could also result in benefit when tumors were already progressing under Herceptin alone. Okay, so we did another phase 2 trial. We finally persuaded the management to provide budget for another phase 2. And we could excite clinicians with the preclinical data we had. And the study was now in HER2-positive metastatic breast cancer patients whose tumors progressed under Herceptin. And we just continued Herceptin treatment and added pertuzumab. There was no chemotherapy. So a really challenging situation, advanced breast cancer progressing under one of the 2 compounds. Now, as I will show the data in a second, as this was quite successful, we amended the trial because the question came up, what can pertuzumab do alone in these patients whose tumor progressed on Herceptin? And as it was not very active, we had patients whose tumor had progressed on Herceptin and then progressed on pertuzumab, and we treated them with a combination again. And here's the results. So, metastatic breast cancer patients whose tumor progressed on trastuzumab, we continued trastuzumab and added pertuzumab. And as you can see, we even saw 5 complete responses. 5 complete responses in advanced stage of breast cancer without chemotherapy. This had never been seen before. This was really amazing. And we had a lot of partial responses. So the overall response rate was 24%. And there was a significant number of disease stabilizations for more than 6 months, and this was also a very important clinical finding because patients were in documented tumor progression when they were enrolled into the study. So the clinical benefit rate was 50%, an amazing result. It was much better than we had expected. So we amended the trial. Now we had patients whose tumors had progressed on Herceptin. We discontinued Herceptin and waited for about 4 weeks or more, and then treated them with pertuzumab alone. And you can see the response rate and the clinical benefit rate was quite low. Now, out of these patients whose tumors had progressed on Herceptin and then on pertuzumab alone, we added the combination. We gave them the combination, and you can see the response rate went up again, and the clinical benefit rate went up again. So this was clear demonstration of synergy of the two compounds. And I personally think that synergy had never been shown so nicely in a clinical trial before. This was really exciting, and what happened was Genentech came back because Genentech had no longer participated in this trial as they gave it up after the 5 negative phase 2 trials. But then they came back, of course. And this is just the explanation of the synergy. Each of the 2 antibodies can do something which the other cannot. So they have complementary modes of action, and this explains the synergy. Now, when Genentech came back, we started to discuss 2 clinical trials. And the first trial was called FRED, because we had a great team leader at the time, and she was very fond of the Flintstones, and Fred Flintstone gave the name for the first study, and it stood for first line research endlessly discussed. The standard of care, Herceptin and docetaxel, compared to Herceptin and docetaxel with pertuzumab. Now, we could not keep the name FRED for this study because the WHO did not accept it, so this study actually became Cleopatra. And you all know the results. It was a very positive trial, both in PFS and in OS terms. So actually, the OS result confirmed that the addition of pertuzumab added more overall survival time to Herceptin and chemotherapy than Herceptin had been able to add to chemotherapy when Herceptin was approved. So this was actually the biggest advance achieved with a single agent in breast cancer treatment. And the combination of Herceptin and Perjeta was approved for treatment of first-line metastatic breast cancer in the USA in 2012 based on the Cleopatra trial. Now, the other study we discussed when Genentech came back was called Barney Rubble. And this was supposed to be a biomarker-assisted study in neoadjuvant therapy. And we at Roche wanted to do this trial for 2 reasons. First of all, a strategic reason, because we thought a neoadjuvant setting is in early breast cancer. And you know, with a neoadjuvant study, you get the result relatively quickly and a relatively clear result. So we thought with a positive result in the neoadjuvant setting, we could embark on an adjuvant trial sooner and would not have to wait for the outcome of the metastatic trial. The other reason why we wanted to do this study was there were still a few scientific questions open. So what could do pertuzumab with chemotherapy? without Herceptin. It had not been tested before. And, of course, in early breast cancer, what can the two antibodies do without chemotherapy? And you can address such questions in a neoadjuvant trial easily without having an ethical problem, because after surgery, patients can have any kind of standard treatment. Now, again, we could not keep this name, Barney, and it became Neosphere. Now, you probably know the outcome here, so the addition of pertuzumab to Herceptin and chemotherapy dramatically increased the PCR rate. And another interesting finding was that there were about 17% of patients in the cohort that received no chemotherapy, but just the two antibodies. So you could say you could actually preclude chemotherapy from these patients. And we did an extensive biomarker program with this study. Unfortunately, we were not able to identify a marker that would predict the patients who do not need chemotherapy. But nevertheless, it was an interesting finding. Now, Genentech had not participated in this trial because at that time, when we designed the trial back in 2007, they were arguing that FDA would not recognize PCR as an endpoint, and therefore the study would not have regulatory purposes. However, based on the outcome of this study, we received first approval for neoadjuvant treatment of early breast cancer with a combination of Perjeta and Herceptin in the USA, although there had not been a single patient from the USA in this trial because Genentech had not participated. So this is also an interesting story with the development of pertuzumab. And of course, the outcome of the Neosphere trial then paved the way for the affinity study, the adjuvant trial of Herceptin and pertuzumab, and you all know that the results were presented last year at ASCO. The trial design was basically standard of care, chemotherapy plus trastuzumab, compared to chemotherapy plus trastuzumab plus pertuzumab. And the treatment was 52 weeks, so for 1 year, and there will be follow-up until 10 years. Now, the result, which we had last year at ASCO, was at a relatively early stage, after 3 years follow-up, and already this result led to the approval of the affinity regimen by the FDA for treatment of early breast cancer patients in the adjuvant setting. And the CHMP opinion was also positive. Actually, on June 1st, so just a few weeks ago, during ASCO time this year, we received also approval from EMA, the European Medical Association, for the combination of Herceptin and Perjeta in the adjuvant setting. And finally, the several international guidelines already recommend the affinity regimen in patients with tumors at high risk of recurrence. So we received both in the USA and EMA the approval for the so-called high-risk patients, which are defined by either lymph node positivity or hormone receptor negative status of the tumors. So this is the current situation. We are very much looking forward to the longer follow-up. The next analysis will be after 5 years, so we are expecting very interesting results in the future, as we also realized that the 3-year analysis was relatively early, and the curves started to separate only after 2 years or so. So this is the interesting story of pertuzumab, and with this I want to close, and maybe there is some questions.
Max, that's an excellent lecture. I want to ask one very simple question. You know the pertuzumab and the trastuzumab in neoadjuvant chemotherapy, the PCR rate, the difference of the PCR rate between the Herceptin alone or Herceptin with pertuzumab, the difference may be 20%. But in affinity study, the adjuvant trial, even the survival benefit is significant, but only 2%. So why the difference between the neoadjuvant and the adjuvant setting? Is there any reason why you can explain to us?
Okay, I think one of the reasons is certainly in the neoadjuvant setting, you treat directly the existing tumor. In the adjuvant setting, you have only, let's say, some circulating tumor cells or micrometastases that are hiding somewhere. And as you can see, if you look at the results, which I did not show today, the curves start to separate only after 2 years. So this means that in the control arm, which receives Herceptin already, the relapses occur relatively late. And I think that the curves will separate more after extended time. So this is, I think, one of the possible explanations. And as you also know, the result of the control arm was unexpectedly good. It's really good news for patients. When we planned the study, we had estimated a progression-free survival rate of 89.2%, and the control arm in this study already had 93%, 93.2%.